in biotechnology, we can see that let's start about this. This is suppose a generalized cell. Generalized cell. And in a generalized cell, this is the nucleus. We know that the nucleus is consisting of what? Nucleus is consisting of chromosomes. And chromosomes are what? Chromosomes are bearers of hereditary characteristic. When we are saying chromosomes, chromosomes are bearers of hereditary characteristic. And yes, Subham. Yes, main accepts of environment we have completed for preliminary examination, and we try to do a balance by covering also some parts of biotechnology right now. Now we can see that these are the bearers of hereditary characteristic, which is called as what? Which is called as chromosomes. Clear? And chromosomes are made up of what? They are made up of DNA. So we say that if suppose chromosomes are molecules, then DNA would be considered as what? DNA would be considered as the atoms. Clear? So yes, we are saying that if chromosomes are molecules, then DNA would be considered as atoms. Fine. Now, fine, Shruti, thank you. But let's cover up this part also. ठीक है, और जहाँ जहाँ हिंदी में जरूरत होगी मैं बोल दूंगा हिंदी में, कोई बात नहीं है. फाइन. क्रोमोसोम्स आर मेड अप ऑफ डीएनए ओनली. सो अगर मान लो कहते हैं कि क्रोमोसोम्स मॉलिक्यूल्स है तो डीएनए क्या है? उसके एटम्स हैं. नाउ डीएनए इज नॉट ओनली कन्फाइंड टू द न्यूक्लियस. डीएनए सिर्फ न्यूक्लियस में नहीं है. डीएनए इज प्रेजेंट इन टू अदर पार्ट्स ऑफ द सेल. दो पार्ट्स में डीएनए और सेल के रहता है. नाउ वन इज कॉल्ड एज प्लास्टिड्स. प्लास्टिड्स देखो प्लांट सेल में रहेंगे, एनिमल सेल में नहीं रहेंगे, ह्यूमंस में नहीं है प्लास्टिड. और माइटोकॉन्ड्रिया, माइटोकॉन्ड्रिया इज प्रेजेंट इन ह्यूमंस आल्सो. माइटोकॉन्ड्रिया इज कॉल्ड एज पावर हाउस ऑफ द सेल. सो वी आर सेइंग that DNA is not only present in nucleus, but it is present in two other parts of the cell. And these two other parts of the cell where DNA is present, these type of DNA would be referred to as what? It is called as extra chromosomal DNA. This is called as what? It is called as extra chromosomal DNA. Thank you, all of you. After biotechnology, we'll take the sleep. We'll say good morning. Let's continue with biotechnology because it will take also time. Fine. Now this is called as what? This is called as extra chromosomal DNA, which is present in, obviously we are talking about a generalized cell in which the characteristic of both animals and plant cell is present. So this would be called as what? It would be called as extra chromosomal DNA. Now, when we are talking about the structure of DNA, the structure of DNA appears to be like this. This is the structure of DNA. Now, this structure of DNA was given by two scientists. Their names were Watson and Crick. And this you have studied in your school days. Watson and Crick. And this was given in 1953. So, this kind of structure of DNA is called as what? It is called as double helix structure. इसे क्या कहते हैं? इसे कहते हैं हम लोग double helix structure of DNA, which was given by Watson and Crick. Clear? And in this structure we can see that nitrogenous bases are present. देखो यहाँ पर nitrogenous base है. Nitrogenous base में क्या क्या है? ATCG, ATCG, ATCG. Now A stands for what? A stands for एडिनाइन. ये आपने पढ़ा होगा स्कूल डेज में. और ए हजम पेयर करेगा टी के साथ, तो टी इज कॉल्ड एज व्हाट? इट्स कॉल्ड एज थाइमाइन, थाइमीन. देन सी स्टैंड्स फॉर व्हाट? सी स्टैंड्स फॉर साइटोसिन. And G stands for what? G stands for guanine. Clear? A stands for adenine, T for thymine, C for cytosine, and G for guanine. Clear? So A always pairs with T. So the pairing of A with T is called as what? It is referred to as, or the pairing of C with G is referred to as what? It is called as base pair. इसे क्या कहते हैं? Base pair. जैसे AT है तो AT is a base pair, CG is a base pair, and the joining of these two bases, दोनों bases जो join हैं, these two bases are joined by what? They are joined by hydrogen bond. We can see hydrogen bond is responsible for joining them. Fine. Hydrogen bond is responsible for joining them. And as far as the strands of DNA are concerned, we can see that in between this sugar unit, phosphate unit is present. Suppose this is sugar unit, sugar इकाई है ये. So in between two sugar unit, a phosphate unit is also present. So this is the structure of DNA. Clear? This happens to be yes, triple parenting etc. would be covered. Don't worry, सब अभी cover करता हूँ. ये सब cover कर देता हूँ शुभम. Don't worry. Yes, mitochondria is yes, fine. I'll try to cover up all these things. Don't worry. Maximum coverage in minimum hour I'll do. Fine. Don't worry about that. Now that's called as what? That's called as base pair ATCG. Now, when we are talking about human genome, जब कहते हैं ना कि human genome, what is human genome? Human genome is referred to as what? It is referred to as the genetic makeup of humans. Genetic makeup of humans. अब देखो ध्यान दो कि humans का genetic makeup किस तरह का है? How is the genetic makeup of human? Clear? How is the genetic makeup of human? Fine. Thank you, Sapna Rathod. Thank you so much. Fine. How is the genetic makeup of humans? Now, when you are talking about genetic makeup of humans, genetic makeup of humans depends upon the sequencing of DNA. See, there is a type of map which is called as what? Which is called as hep map. एक map होता है जिसे हम लोग कहते हैं क्या? इसे कहते हैं hep map. And hep map is a type of map which compares the genetic makeup of different organisms. अलग अलग organism के genetic makeup को compare करता है. So that is called as what? That is called as hep map. लेकिन genetic makeup में compare किसे कहते हैं? See, मान लो कि humans हैं, तो humans का genetic makeup है. DNA को हम लोग examine करते हैं और DNA को देखते हैं कि what is the sequencing of base pairs. तो sequencing of base pairs का मतलब है कि किस तरह से base pairs sequenced हैं, किस तरह से base pairs arranged हैं. Clear? So we are saying that sequencing of base pairs, किस तरह से base pairs sequenced हैं, किस तरह से वो arrange है. मान लो एक sequence है, there's one sequence and that is ATCG. Another sequence can be another ATCG. So there are different sequences. So when we are saying that a sequencing of base pair is examined, we can see that sequencing of base pair would be examined in human genome. So human genome, when we say genetic makeup of humans, so what are we responsible for examining of humans? We are responsible for examining of humans the sequencing of base pairs, that how the base pairs are arranged. Clear? How the base pairs are arranged. But human genome is referred to as what? Human genome is referred to as book of life. इसे क्या कहते हैं? Book of life. Human genome is called as blueprint of life. Human genome includes what? It includes genes. See, when we say that when we say chromosomes are the bearers of hereditary characteristic, they are made up of DNA. Then what is genes? Genes are nothing but parts of DNA only which would be responsible for. You can refer any book. I won't say so. You can refer any book of your choice. But now, when we are talking about human genome, we say that human genome are what? Human genome are genetic makeup of humans. This is called as what? This is called as book of life. This is called as what? Blueprint of life. And human genome consists of what? It consists of genes, their variants, and also regulatory genes which are responsible for controlling other genes. तो क्या हुआ? इसमें क्या क्या है? उसमें है genes भी. Genes क्या है? Genes are nothing but parts of DNA only which are responsible for reflecting different kinds of characteristic. Genes क्या है? Genes are nothing but parts of DNA only which are responsible for reflecting different kinds of characteristics. So these are called as genes. Responsible for reflecting different kinds of characteristic. And as far as see, you can read any book you want, whichever you feel comfortable you can read. Fine. No, not any link with biocomputers as such here. Now see, now they are responsible for what? हम लोग कहते हैं कि ह्यूमन जीनोम को कहते हैं बुक ऑफ लाइफ, ह्यूमन जीनोम को कहते हैं ब्लूप्रिंट ऑफ लाइफ. तो ऑब्जेक्टिव टाइप क्वेश्चंस इस तरह से आ सकते हैं. लेकिन ह्यूमन जीनोम में है क्या? ह्यूमन जीनोम में है जीन्स हैं, उनके वेरिएंट्स भी हैं, मतलब उनके अपरूप भी हैं, एंड रेगुलेटरी जीन्स, कुछ ऐसे जीन्स हैं जो दूसरे जीन को कंट्रोल करते हैं, एंड रेगुलेटरी जीन्स विच आर रिस्पॉन्सिबल फॉर कंट्रोलिंग अदर जीन्स. सो ह्यूमन जीनोम कंसिस्ट ऑफ जीन्स, देयर वेरिएंट्स, एंड रेगुलेटरी जीन्स विच आर रिस्पॉन्सिबल फॉर कंट्रोलिंग अदर जीन्स. नाउ ह्यूमन जीनोम वाज डिसिफर्ड व्हेन? It was deciphered in the year 2000, and it was deciphered in the year 2000 by the scientists of Europe, and a private company, and that private company is called as what? It is called as Celera Genomics. Clear? So it was deciphered, and it took roughly about 7,50,000 A4 sheets to print the entire genetic makeup of humans. The difference between genes and chromosome is what? See, chromosomes are what? Chromosomes are bearers of hereditary characteristic. मतलब कि hereditary characteristic को हेरिडिटरी कैरेक्टरिस्टिक को एक जनरेशन से दूसरे जनरेशन तक ले जाएगा. सो दे आर बियरर्स ऑफ हेरिडिटरी कैरेक्टरिस्टिक, एंड देन वी कैन सी that they are bearers of hereditary characteristic, and also we can see that as far as human genome is concerned, देखो क्रोमोसोम्स को कहते हैं bearers of hereditary characteristics, जो आपके न्यूक्लियस में है. क्रोमोसोम बना किसका हुआ है? डीएनए का. It is made up of what? It is made up of DNA. And what is genes? Genes are nothing but parts of DNA only which are responsible for reflecting different kinds of characteristic. Genes are parts of DNA only which are responsible for reflecting different kinds of characteristic. Clear? Responsible for reflecting different kinds of characteristic. चलो, ये हुआ एक चीज. Now देखो, ह्यूमन जीनोम वॉज डिसिफर्ड इन द ईयर 2000. ये देखा गया कि किस तरह का ह्यूमन जीनोम दिखता है. सो इट वॉज डिसिफर्ड इन द ईयर 2000, एंड इट वॉज डिसिफर्ड बाय द साइंटिस्ट ऑफ यूरोप एंड अ प्राइवेट कंपनी कॉल्ड सेलेरा जेनोमिक्स, एंड इट took roughly about 7,50,000 A4 sheets to print the entire genetic makeup of humans. इतने sheets लगे थे genetic makeup of humans को print करने में. Clear? It took roughly about this much sheets to print the entire genetic makeup of humans. So that's called as human genome. Fine. But as far as human genome is concerned, we can see that human genome is also referred to as what? It is referred to as book of life, blueprint of life. ये सब तो मैंने आपको बता दिया. But earlier the perception of scientists was what? That there are 3 billion base pairs in human cell. And these 3 billion base pairs are roughly responsible for constituting 1 lakh genes. But after the human genome, we came to the conclusion that only 20,000 genes are present in humans. Clear? Oh, only 20,000 genes are present in humans and not 1 lakh as thought earlier. Now, in human genome, we are responsible for determining what? In human genome, we are responsible for determining the sequencing of base pairs. किस तरह से base pairs के sequence है? AT, मान लो CG, AT, ये determine करते हैं. But the same sequencing of base pair is determined in case of what? It is determined in case of DNA fingerprinting also. उसी तरह से DNA fingerprinting में भी determine करते हैं हम लोग sequencing of base pairs. The same sequencing is being determined. So what is the difference between human genome and DNA fingerprinting then? तो difference क्या है human genome में? उसी sequencing को हम लोग देखते हैं human genome में difference है. देखो, more than 99% of genes among us is exactly the same. Yes, you may use that while you are teaching. Now, more than 99% of genes among us is exactly the same. हम लोग में, सारे में, हम लोग में 99% से genes एकदम एक ही जैसे हैं, exactly the same. And if suppose we are responsible for studying the sequencing, अब genes क्या है? Genes DNA ही है. So अगर मान लो sequencing पढ़ा जाए more than 99% of genes, क्योंकि DNA है तो उसमें sequencing होगा. जीन्स अगर है तो डीएनए है, डीएनए अगर है तो उनमें सीक्वेंसिंग होगा. सो इफ वी आर रिस्पॉन्सिबल फॉर स्टडिंग द सीक्वेंसिंग ऑफ मोर देन 99% ऑफ द जीन विच इज कॉमन अमॉन्ग अस, it is called as human genome. And if suppose we are responsible for studying the sequencing of less than 1% of the gene which is uncommon among us, then it is called as what? Then it is called as DNA fingerprinting. Clear? DNA fingerprinting. Fine. And where? Now, लेकिन ये बात होती है कि कहाँ अनकॉमन कहाँ है. तो मान लो ये लंबा चौड़ा ह्यूमन जीनोम है, 7,20,000 A4 शीट्स लगे थे इसको प्रिंट करने में. दिस इज द ह्यूमन जीनोम. Now we can see there are some sequence of base pairs which are repeated at frequent intervals. There are sequence of base pairs which are repeated at frequent intervals. These sequence of base pairs which are repeated at frequent intervals are called as what? It's called as STR. STR means short tandem repeats. And it is here that we differ from each other. यहाँ पर हम एक दूसरे से भिन्न हैं. So here we are responsible for differing from each other. We are different from each other. So your STR may be resembling your mother and father, but won't be exactly the same. अगर मान लो STR भी same हो गया तो you are the clone of your mother and father. Clear? But it won't be exactly the same. But here we are saying that STR is that where we differ from each other. Otherwise, we are exactly the same. Clear? This is called as DNA. ये DNA fingerprinting और human genome के बीच में अंतर है. लेकिन the thing is that why we were responsible for conducting human genome in the year, why we are responsible for deciphering the human genome in the year 2000. The objective was what? The objective was to find the place of humans on the HEP map. Where are humans located on the HEP map? Clear? First. Then also the objective was what? The objective was not only to determine the place of humans on the HEP map. The objective was also that there are diseases related with the genetic makeup of humans. In order to find remedy for these diseases, the deciphering of human genome was required. Clear? जो हमारे जेनेटिक मेकअप हैं उनसे क्या है कि डिजीजेस एसोसिएटेड है, तो उन डिजीजों के रेमेडी के लिए भी हमें ज़रूरी था ह्यूमन जीनोम को डेसिफर करना. And also we can see that as far as human genome is concerned, the objective was also to that it would be helping in organ transplant. Organ transplant कैसे करते हो? Organ transplant करते हो कि दूसरे एनिमल्स से ऑर्गन को लेकर मान लो ह्यूमन में ट्रांसप्लांट किया, तो इफ वी डू नॉट नो Thank you so much, Rakesh. Like after 10 years, now I understand the real meaning of HDR. माइक्रो इतने रात तक तो कभी नहीं पढ़ा. Very good, very good. सर प्लीज डिस्कस लिटिल बिट अबाउट हेप मैप. क्या है ना, हेप मैप इज़ अ मैप जिसमें हम लोग क्या करते हैं कि जेनेटिक मेकअप अलग अलग ऑर्गेनिजम्स का कंपेयर करते हैं और देखते हैं कि कौन किसके क्लोज है. क्लियर? जैसे हम लोग से कौन क्लोज है बच्चों, हम लोग से अगर बच्चों इस तरह से आप पढ़ते रह जाओगे, दिन में पढ़ते अभी सो गए तो पीछे हो जाएंगे क्या आप लोग से. देखो, अगर आप 10 दिन 15 दिन भी इस तरह से पढ़ लिए तो पीटी तो आपके पॉकेट में है, क्योंकि इसमें क्या होता है, एफर्ट लगता है बच्चों, और एफर्ट लगता है तो याद भी रहता है आपको. मैं तो कहता हूं कि जो कन्वेंशनल जो आपके स्टैटिक पोर्शन है, हिस्ट्री, ज्योग्राफी, पॉलिटी, उसको एक बार जबरदस्त रूप से ऐसे ही पढ़ लीजिए. क्लियर? और देखिए क्या डिफरेंस है. मुझे लगता है कि डिफरेंस हो जाएगा फिर प्रिलिमिनरी एग्जामिनेशन में, और अभी समय है उसे कर सकते हैं. वो सर पे रह जाएगा प्रिलिमिनरी एग्जामिनेशन तो कर नहीं सकेंगे, तो एक दो दिन जरा जम कर लगा दें उसमें. फाइन. नाउ सी, as far as human genome is concerned, we can see the purpose of human genome was what? To determine the place of humans on the HEP map. तो एक तो ये है. Second is it would help in organ transplant. Third is there are diseases associated with the genetic makeup of humans. और मैंने कहा हेप मैप का मतलब है क्या, कि हेप मैप इज अ मैप विच कंपेयर्स द जेनेटिक मेकअप ऑफ डिफरेंट ऑर्गेनिज्म, और उसमें ये देखना है कि वेयर इज द प्लेस ऑफ ह्यूमंस ऑन द हेप मैप. ह्यूमंस का कहाँ है जगह है हेप मैप पे. क्लियर? व्हाट इज द प्लेस ऑफ ह्यूमंस ऑन द हेप मैप. सो ऑल दीज थिंग्स नीड्स टू बी डिटरमाइंड. हम लोगों के सबसे निकट कौन है? हम लोगों के सबसे निकट है चिंपांजीज. ह्यूमंस के सबसे निकट चिंपांजीज. चिंपांजीज के जीनोम में देखिएगा तो सर्टेन गैप्स है, जो इन ह्यूमन दे आर फिल्ड अप विद अ डीएनए विच इज कॉल्ड ए जंक डीएनए. सो जंक डीएनए इज द ओनली डिफरेंस दैट वी हैव विद चिंपांजीज. देखो अर्लियर क्या था कि साइंटिस्ट को लगता था कि जंक डीएनए का कोई इंपॉर्टेंस ही नहीं है, सिग्निफिकेंस ही नहीं है, तो उन्होंने उसे नाम दे दिया जंक. अब उन्हें पता चल रहा है कि जंक डीएनए के कारण ही हम ह्यूमंस हैं, अदरवाइज जंक डीएनए अगर हट जाए तो what can we transform into? We can transform into chimpanzees also. Fine. So the junk DNA is. But when we talk about DNA fingerprinting, मान लो जब DNA fingerprinting कहते हैं तो DNA fingerprinting is individual identification of an individual. अलग यूनिक आइडेंटिफिकेशन जो है इंडिविजुअल का, वो DNA fingerprinting मानते हैं. लेकिन अभी देखो क्या हुआ, 2-3 साल पहले पेटर्निटी टेस्ट कंडक्ट किया गया था. पेटर्निटी टेस्ट कंडक्ट किया गया था एक यूएस सिटीजन पर. तो पेटर्निटी टेस्ट का मतलब है कि क्या वह व्यक्ति अपने बच्चे का ऑनेस्टली फादर है कि नहीं. सो दैट्स कॉल्ड एज पेटर्निटी टेस्ट. एंड व्हाट हैपेंड वाज दैट ऑल्दो द पर्सन वाज ऑनेस्टली द फादर ऑफ हिज ओन सन, बट ही फेल्ड द पेटर्निटी टेस्ट. और दिमाग में दौड़ाना तुम लोग ज़्यादा. he failed the paternity test and it was found that the child belongs to his uncle. और भी मत दिमाग दौड़ाना. बट द प्रॉब्लम वॉज द अंकल वॉज नेवर बॉर्न. अब ये क्या बात कर रहे हैं हम लोग, व्हाट वी आर डिस्कसिंग. सो वी आर डिस्कसिंग एक्सेप्शंस टू डीएनए फिंगरप्रिंटिंग. देखो डीएनए फिंगरप्रिंटिंग तो कई बार आपके एग्जामिनेशन में पूछा गया है. दे हैव आस्क्ड यू नंबर ऑफ टाइम्स डीएनए फिंगरप्रिंटिंग इन योर एग्जामिनेशन. बट honestly he was the father of his own son, but it was found that biologically the son belongs to the uncle and the uncle was never born. That is also a reality. So let us find out where the uncle was and let's discuss the exceptions of DNA fingerprinting. Although DNA fingerprinting means what? DNA fingerprinting means that's unique identification of an individual, and this was developed by Mr. Alec Jaffrey. So DNA fingerprinting, we can see, is a unique identification of an individual. Unique identification of an individual. And this DNA fingerprinting was developed by Alec Jaffrey. Alec Jaffrey. Clear? And how is DNA fingerprinting conducted? First of all, a biological sample would be taken. तो biological sample कुछ भी ले सकते हैं. It can be blood, which can be taken as the biological sample. तो blood ले लो biological sample के लिए. But remember that in blood, only WBC would be having DNA. RBC में DNA नहीं रहता है. So only WBC has DNA. RBC does not have DNA present in it. Clear? So the biological sample was taken, and one drop of blood was now what we do is that from the blood we are responsible for. मान लो कि WBC है, तो इस ब्लड से क्या करते हैं कि एक पोर्शन DNA का कट करते हैं. मान लो इसमें DNA है तो एक पोर्शन DNA का काटते हैं. और किस पोर्शन को काटते हैं आप जानते हो? STR, that is short tandem repeat. When you are cutting this portion of DNA, अंकुर सिंह तो कुछ टॉपिक्स हैं जैसे माइटोकॉन्ड्रियल डीएनए ट्रांसफर डिस्कस करेंगे और क्लोनिंग डिस्कस करेंगे, थोड़े से स्टेम सेल्स को भी डिस्कस कर लेंगे, जीएम मस्टर्ड थोड़ा न्यूज़ में है उसको डिस्कस कर लेंगे, कुछ टॉपिक्स कर लेते हैं आज, ठीक है? तो मुझे लगता है कि ये 3 बजे तक ख़त्म हो जाएगा, 3 साढ़े 3 बजे तक. उसके बाद यू कैन स्लीप. आई विल ऑल्सो टेक रेस्ट. फाइन. नसीब पहले क्या करते हैं, डीएनए सैंपल लिया, बायोलॉजिकल सैंपल लिया, और ये ब्लड हो सकता है. तो ब्लड से क्या कर रहे हैं, WB से डीएनए को एक्सट्रैक्ट किए और डीएनए के एक पोर्शन को कट किया, और इसको कट किया किससे? STR से. किस पोर्शन को कट किया? STR को. STR वो है जहां पर हम लोग एक दूसरे से अलग हैं. Now this would be cut with the help of what? This is cut with the help of restriction enzyme. Restriction enzyme. Restriction enzyme behaves as a molecular seizure. So this we are cutting with the help of restriction enzyme. Fine. And what we are doing is that not only restriction enzyme, on this we are responsible for conducting a test. And this test is called as what? This test is called as southern blotting test. इस पर क्या करते हैं? एक टेस्ट कंडक्ट करते हैं. And that test is called as what? Southern blotting test. हाँ, थोड़ी देर और करेंगे, 3 घंटे तक लेते हैं बच्चों इसको खत्म, 2.5 घंटे तक और है बस, उसके बाद खत्म हो जाएगा. 2 घंटे 2.5 घंटे और लेता हूँ, ये खत्म हो जाता है कुछ टॉपिक, तो उसके बाद मैं भी रेस्ट करूँगा, आप भी रेस्ट करो. नाउ दिस इज कॉल्ड एज, इस STR को जो सैंपल है डीएनए का, जब आप लोग इसे काटते हो तो उस समय क्या है ना, उस समय ये सीक्वेंसिंग और बेस पेयर नज़र नहीं आता है. बट व्हेन यू कंडक्ट अ टेस्ट विच इज कॉल्ड एज व्हाट? विच इज कॉल्ड एज सदर्न ब्लॉटिंग टेस्ट. Blotting test. Then we can see sequencing of base pairs can be visible. Then sequencing of base pair is visible. So it is said that DNA fingerprinting has been conducted. But as far as DNA fingerprinting is concerned, the objective of conducting DNA fingerprinting is to identify dead person. Clear? Dead person को identify करते हैं इससे. Which we are also responsible for identifying what? Not only a dead person, we also identify a criminal. We also identify a dead person, biological parents also is identified. This is the purpose. But today we can see that the Supreme Court has said now that DNA fingerprinting cannot be considered as foolproof identification of an individual. क्यों? Why cannot DNA fingerprinting be considered as foolproof identification of an individual? Why is that? Because there is a term called human chimera. अब देखो, एक टर्म है जिसको हम लोग कहते हैं human chimera. Human chimera का मतलब है a person having more than one DNA fingerprint. एक ऐसे व्यक्ति जिसमें एक से ज़्यादा DNA फिंगरप्रिंट हो. तो यूपीएससी में कई बार आपको पूछा है डीएनए फिंगरप्रिंटिंग, हो सकता है नहीं दे रहा है, लेकिन ह्यूमन कीमेरा तो पूछना बनता है ना, बिकॉज अ पर्सन हैविंग मोर देन वन डीएनए फिंगरप्रिंट. नाउ लेट्स सी दैट हाउ ह्यूमन कीमेरा इज पॉसिबल. किस तरह से ह्यूमन कीमेरा पॉसिबल है? नाउ ह्यूमन कीमेरा इज पॉसिबल हाउ? बिकॉज यू वुड हैव सीन ट्विंस. ह्यूमन कीमेरा का मतलब है क्या कि एक ऐसा व्यक्ति जिसमें एक से अधिक डीएनए फिंगरप्रिंट हो, अ पर्सन हैविंग मोर देन वन डीएनए फिंगरप्रिंट एज सच, क्लियर, वुड बी कॉल्ड एज ह्यूमन कीमेरा, बिकॉज कीमेरा इज एन ऑर्गेनिज्म विच हैज मोर देन वन जेनेटिक मेकअप, clear, more than one genetic makeup, chimera. And you would have seen twins. Some twins are called as identical twins. Identical twins, they look like, they are also called as monozygotic twins. Monozygotic twins. So they are similar, they look like, but monozygotic twins are formed how? See, suppose this is a zygote which has been formed after fertilization of the egg of the mother with the sperm of father. Sometimes what happens, that zygote undergoes a division and two identical zygote will be formed. Now this leads to the formation of identical twin or monozygotic twin. So we can see that this leads to the formation of identical twins or monozygotic twins. And generally monozygotic twins are suspended in different amniotic sac and they do not share the same placenta. देखो ये जो fetuses है ना monozygotic twins के, identical twins के, वो एक साथ नहीं रहते हैं जनरली, वो अलग अलग amniotic sac, ये जो थैली है अलग होगी. This is called as what? This is called as amniotic sac. And this is called as placenta. Placenta का मतलब umbilical cord through which nourishment is given to the baby. Placenta or umbilical cord through which nourishment is given to the baby. So you can see that generally these twins are present in different amniotic sac. They are not sharing the same placenta. Fine. And they lead to the birth of identical twins. They look similar. But sometimes, see, when we talk about the womb of the mother, this is the womb of the mother, this is the fetus, this is the umbilical cord, which is also called as placenta. Inside the womb, there is a sac also, which is called as amniotic sac. And inside the womb also we can see a fluid is present, which is called as what? Which is called as amniotic fluid. Amniotic fluid. Clear? And sometimes if suppose the mother feels discomfort, a hollow needle is inserted inside the womb and some of the amniotic fluid would be withdrawn. And this amniotic fluid which is withdrawn consists of, yes, I am coming to Krishna Oja, I am coming to that. When we are talking about amniotic fluid is withdrawn, and this amniotic fluid is examined, because amniotic fluid consists of what? It consists of the cells of the fetus. Fetus के सेल उसमें रहते हैं, तो एग्जामिन करते हैं कि उसमें अगर किसी तरह की जेनेटिक डिसऑर्डर हो कि नहीं, है कि नहीं. And this process is called as what? This process of taking amniotic fluid and determining the cells genetically is called as what? It's called as amniocentesis. इसको क्या कहते हैं? Amniocentesis. Clear? This is referred to as amniocentesis. Fine. Amniocentesis, that is the purpose. Sometimes what can happen, that the fetuses leading to the birth of identical twins may be present in the same amniotic sac and they would be sharing the same placenta. Clear? This is a rare case of identical twin. Rare case of identical twins. These kind of twins, identical twins, are called as what? It's called as mono twins. इसे क्या कहते हैं? Mono twins. Generally, when we talk about identical twins, they have same DNA fingerprinting. Why? Because they are result of same fertilization. Only division in zygote has taken place. Otherwise, they are the same. Only division in the zygote has taken place. Otherwise, they happen to be the same. So they are the result of what? They are the result of सिंगल फर्टिलाइजेशन, एक ही फर्टिलाइजेशन से डिवीजन होता है जायगोट में, तब आइडेंटिकल ट्विंस बनते हैं. So they have the same genetic makeup, but we can see एमनियोटिक फ्लूइड में, तो एमनियोटिक फ्लूइड में और जायगोट में, जायगोट क्या है, फीटस जो है, तो फीटस में जो डीएनए है, एमनियोटिक फ्लूइड के जो सेल में डीएनए है वही फीटस का डीएनए है. क्लियर? नाउ सी, एक ट्विन होता है जिसको हम लोग कहते हैं फ्रेटर्नल ट्विन, उसको कहते हैं डाइजाइगोटिक ट्विन. क्लियर? फ्रेटर्नल ट्विन का मतलब है कि ये तो हो गया आइडेंटिकल ट्विन्स के केस, लेकिन फ्रेटर्नल ट्विन्स में क्या है कि द मदर इज रिस्पॉन्सिबल फॉर रिलीजिंग मोर देन वन ओवम एट अ टाइम. और ये जो ओवम है वो फर्टिलाइज हो जाएंगे फ्रॉम दी स्पर्म ऑफ फादर. तो मान लो अगर 2 ओवम मदर ने रिलीज किया है, तो they would be fertilized by the sperm of father, and that would be resulting in the formation of what? That would be resulting in the formation of 2 zygote. But these 2 zygote are separate and the fertilization is different. Clear? And the fertilization is different. So you can see, if the fertilization is different, they are having different genetic makeup. Their DNA fingerprinting is different. Clear? Their DNA fingerprinting is different. They are having different genetic makeup as such. And when they would be born, they would be just like brothers and sisters. They would be present in the womb, separate amniotic sac. But when they are born, they are just like brothers or sisters. My genetic makeup is not like my, now our genetic makeup, my genetic makeup may resemble my biological parents. And my brother's genetic makeup also may resemble my biological parents. But my genetic makeup and my brother's genetic makeup won't be identical, because we are the result of different fertilizations. We are the result of different fertilizations. So sometimes what can happen? The fetuses which is leading to the birth of fraternal twins, if they are present in the same amniotic sac and they share the same placenta, what will happen? Exchange of cells between them can take place through amniotic fluid. We are saying that exchange of cells between them can take place through amniotic fluid. Amniotic fluid. Clear? So, because you know that the cells of the fetus is present in the amniotic fluid. Suppose this is A, this is B. So, cells of A also present, cells of B also present. So, the cells of both would be present in the amniotic fluid, and what will happen? And what will happen? All mammals have same mechanism, yes. Now, what will happen is that during pregnancy, the cells of each other would be absorbed by each other's body. Clear? जब प्रेगनेंसी हो रहा है तो उसी समय द सेल्स, जैसे यहाँ पर है, मान लो कि दोनों एक ही एमनियोटिक सैक में हैं, एक ही प्लेसेंटा को शेयर कर रहे हैं, तो सेल्स ऑफ ईच अदर वुड बी अब्सॉर्ब्ड बाय ईच अदर्स बॉडी. दैट दिस वुड लीड टू मोर देन वन जेनेटिक मेकअप इन द फिटसेस. इससे क्या होगा? देयर वुड बी मोर देन वन जेनेटिक मेकअप इन द फिटसेस, एंड the baby that is born would be having more than one genetic makeup, and it would be called as what? It would be referred to as human chimera, because the baby is having, the child is having one genetic makeup of his own and one of his fraternal brother or sister, whatever may be the case. Clear? So this is and sometimes what can happen? Sometimes the zygote which is leading to the birth of fraternal twins can fuse with one another and develop as a single zygote. So here was the father's zygote in which the uncle's zygote fused. So uncle was never born, but when the father reproduced, the reproductive cells of the father had the genetic makeup of uncle. That's why the paternity test of the person failed, although he was the actual father of his son. And this type of, so what we can see is that in this case, absorption of cell is responsible for more than one genetic makeup. So if absorption of cell is responsible for more than one genetic makeup in humans, this process is called as what? This process is called as chimerism. I am not understanding anything, sir. शहरी सिराज, क्या नहीं अंडरस्टैंड कर रहे हो बच्चों, बोलो. Because I study in class 8. Oh my God. Oh my God. Fine. Then grow up then to understand these things. Now this is called as what? This is called as chimerism. So we are saying that if absorption of cell is responsible for multitude of genome in humans, more than one genetic makeup in humans, more than one DNA fingerprinting in humans, this process is called as what? It is called as chimerism. And the person would be called as what? Human chimera. A person having more than one DNA fingerprint. Clear? A person having more than one DNA fingerprint. Fine. I will make you understand. Fine. Now this is called as chimerism. But now let us discuss also some other things. See, a question which is asked generally in your examination is also about what? Also about triple parent baby. Now suppose fertilization takes place. We know that fertilization takes place, and in fertilization, this is suppose the nucleus of the mother, this is the egg of the mother. Egg of the mother. And this has been fertilized by sperm of father. So the genetic material of the father, and this is the mitochondria of the mother. Mitochondria of the mother. Fine. And this is the nucleus of the mother. So when fertilization occurs, the genetic material of the father only reaches the nucleus, where father's genetic material is present and mother's genetic material is present. But it does not reach the mitochondria. And you know that DNA is present in mitochondria. So the DNA which is present in mitochondria is of the mother. Clear? When fertilization takes place, we can see when fertilization takes place, we can see that DNA which is coming from the sperm is reaching only and only the nucleus of the egg. It is never reaching the mitochondria. Clear? But mitochondria is having DNA of the mother only. So the conclusion is what? That the cells that we have in our body, the mitochondrial DNA belongs to our mother. It does not belong to our father. The mitochondrial DNA belongs to our mother. It does not belong to our father. Clear? And if we say that 20,000 genes are present in humans, out of the 20,000 genes, only 37 genes are determined by mitochondrial DNA. 37 genes are determined by mitochondrial DNA. The rest is not at all determined by mitochondrial DNA. Clear? The rest is not at all determined by mitochondrial. Only 37 of the genes are determined by mitochondrial DNA. The rest is not determined by mitochondrial DNA as such. Clear? And these would be responsible for what? These would be responsible for sometimes diseases. Clear? So they would be responsible for sometimes diseases. So mitochondrial DNA may be associated with disorders. Mitochondrial DNA may be associated with disorders. So if suppose this mitochondrial DNA which is present in mother's mitochondria is responsible for disorder, now it would be directly transferred from the mother to the child. मदर में ये डिसऑर्डर होगा ही, क्योंकि माइटोकॉन्ड्रिया मदर का है, लेकिन जब मदर रिप्रोड्यूस करेगी तो वो डिसऑर्डर चला जाएगा चाइल्ड में भी. लेकिन एक बात बताओ, अगर मान लो कि कोई मदर ये जानती हो कि उसमें जो डिसऑर्डर है वो उसके बच्चे में जाने वाला है, इज गोइंग टू हिज ओन चाइल्ड, शी वोंट विश टू अंडरगो प्रेगनेंसी. वो प्रेग्नेंट होना ही नहीं चाहेगी. क्लियर? शी वोंट विश टू अंडरगो प्रेगनेंसी, एंड दिस इज नॉट गुड. So she should have the right to have a child. She should have the right to have a child. So for this purpose, triple parent baby is proposed. So what we are doing is that is suppose this mitochondrial DNA, this mitochondria is associated with disorders. So what we do is we mechanically remove the nucleus from the fertilized egg. तो इससे क्या करते हैं, मैकेनिकली न्यूक्लियस रिमूव कर लेते हैं, जिसमें जेनेटिक मटेरियल फादर का भी है और जेनेटिक मटेरियल मदर का भी है. क्लियर? जेनेटिक मटेरियल फादर का भी है और जेनेटिक मटेरियल मदर का भी है. तो इससे न्यूक्लियस से रिमूव कर लेते हैं. एंड देन व्हाट वी डू इज वी आर रिस्पॉन्सिबल फॉर थ्रोइंग अवे द सेल. We do what? We throw away, discard the cell. Responsible for discarding the cell, throw away कर देते हैं सेल को. Now what we do is select another mother. दूसरे मदर को देखते हैं. Now see, her egg cell would be selected. First of all, this would be examined whether the her egg cell has proper mitochondrial DNA. मान लो इसे M1 कर देते हैं. And the nucleus of her egg would be removed. इसे निकाल देंगे. So the nucleus of the fertilized egg would be transferred. So ultimately what we have in the egg is that the genetic material of father and genetic material of the mother. But also we can see that the mitochondrial DNA belongs to the second mother. In the nucleus we have genetic material of the father and genetic material of the first mother. But mitochondrial DNA belongs to the second mother. Now this would be now planted inside the womb of surrogate mother. Surrogate mother के womb में plant करेंगे. The baby which develops would be free from mitochondrial DNA disorders. उसमें mitochondrial DNA का disorder नहीं होगा. But would be having what? Would be having one father and two mother. एक फादर हैं और दो मदर हैं. इसको क्या कहते हैं? इसको हम लोग कहते हैं ट्रिपल पेरेंट बेबी. ट्रिपल पेरेंट बेबी. इज दैट क्लियर? नाउ दिस इज कॉल्ड एज ट्रिपल पेरेंट बेबी. सो दिस इज हाउ ट्रिपल पेरेंट बेबी इज फॉर्म्ड. इज दैट क्लियर? दिस इज हाउ ट्रिपल पेरेंट बेबी इज फॉर्म्ड. Now this is not, is this gene editing? No, this is not gene editing. And now what is surrogate mother is a mother which is responsible for lending her womb only. And I think that's clear to you. Fine. This is called as triple parent baby. अब देखो, एक होता है जिसे हम लोग कहते हैं क्लोनिंग. ट्रिपल पेरेंट बेबी is based on what? It is based on mitochondrial DNA transfer. See, you cannot remove the mitochondria from the egg. If you remove the mitochondria from the egg would die. Why? It's called as the powerhouse of the cell. So it is in this manner only that mitochondrial DNA can be transferred. So this is called as triple parent baby. Now see. Now there's another thing that you have to know, and that's called as cloning. Cloning means clone of an organism is an exact replica of an organism. Clear? Which is genetically and physically similar to the donor. So when you are talking about clone is an exact replica of an organism which is genetically and physically similar to the donor. मेरी आवाज में दर्द आ गया है? ओके. मेरी आवाज में दर्द नहीं आया बच्चों. अभी मैं 1-2 घंटे तो आराम से 2 घंटे तो आराम से खींचूंगा, उसके बाद जाकर घर में सो जाऊंगा. क्लियर? आप चिंता न करें, ये पढ़ लें आप अच्छी तरह से. हाँ, अब वो yes, what a great resilience. Thank you so much. अच्छे बच्चे हैं आप लोग, आप सब ज़बरदस्त रूप से मेहनत करें और पीटी को क्लियर कर लें। सबसे बड़ी रिवॉर्ड मेरी वही होगी। If suppose you clear your examination, then certainly that would be the biggest reward that I'll get. See, we are teachers in different institutes. Teachers teach and they are satisfied with their teaching, but the biggest reward for a teacher is when सर इंस्टीट्यूशंस मैत्री रुक जाओ। कुछ इंस्टीट्यूशंस मैत्री खुद से तो पढ़ लो, इतने तो चीज़ हमने पढ़ाए आपको। इंस्टीट्यूशंस तो फैक्चुअल है। फ्रॉम KSG बैंगलोर, ओके, हाँ हाँ, getting reward from my side, yes, let's see what reward you give. Great to see you after KOL once again, thanks for our effort. Nandini, can we refer attached PDF for current affairs coverage? Yes, you can do anything. Okay, मैत्री बस नाम बता दीजिए। देखो नाम में क्या है, देखो कुछ नाम ले लो इसमें। एक तो साइट्स कर लो, IUCN कर लो थोड़ा सा, कुछ ऑर्गेनाइजेशंस कर लो जैसे बॉम्बे नेचुरल हिस्ट्री सोसाइटी, बर्ड लाइफ इंटरनेशनल, ट्री पर एक ऑर्गेनाइजेशन है उसे कर लो। दिस इज नो मेथड टू रिप्लेस माइटोकॉन्ड्रिया। Yes, Krishna Oja from KSG. Oh my God, West Bengal. Very good. Questions I'll send. Tomorrow I'll tell them. All of them are fast asleep now. So tomorrow I'll make sure that these questions are, whatever the questions we have discussed in the class would be sent to you. Clear? Don't be concerned about that. I'll send the questions. Yes, I'll attach. न्यूक्लियस में डीएनए कहाँ होता है? न्यूक्लियस में डीएनए होता है क्रोमोसोम्स में। न्यूक्लियस में होते हैं क्रोमोसोम्स, क्रोमोसोम्स डीएनए से ही बने हुए होते हैं। और क्रोमोसोम डीएनए और कहाँ रहता है? इट इज प्रेजेंट इन प्लास्टिड ऑल्सो एंड इट इज प्रेजेंट इन माइटोकॉन्ड्रिया ऑल्सो। क्लियर? प्लास्टिड ऑल्सो एंड इट्स प्रेजेंट इन माइटोकॉन्ड्रिया ऑल्सो। अब देखो क्लोनिंग, इसे थोड़ा सा समझो। क्लोनिंग मीन्स, क्लोन मीन्स एन आइडेंटिकल रेप्लिका, and this is genetically and physically similar to the donor. दोनों होगा, genetically भी होगा similar to the donor and physically similar to the donor. And as far as cloning is concerned, the first successful experiment of cloning was conducted in 1996. And this was conducted at Roslin Institute, Edinburgh. Clear? And cloning is based on what? It is based on somatic cell nucleus transfer. Somatic cell nucleus transfer. Somatic cell nucleus transfer. Clear? तो ये थोड़ा सा जानना है, somatic cell nucleus transfer. Then we can see that what is somatic cell nucleus transfer and how cloning is conducted. Let's understand, because we are saying that it is the replica of an organism. Clone is a replica of an organism which is genetically and physically the same. Now on one side we are taking what? We are taking the donor. Donor is the person whose clone is to be prepared, and from him we are extracting what? We are extracting some somatic cells. Somatic cells are extracted. Now when we extract somatic cells, we are responsible for putting these cells in quiescent stage. Clear? Dolly dolly. Yes, Maitri, dolly dolly. Now we are responsible for putting this in the quiescent stage. Quiescent stage. Quiescent stage means where the cell division stops. And we are responsible for giving them chemicals so that they revert back in their life cycle. So they are reverting back in their life cycle. reverting back in their life cycle. And life cycle means each and every cell would be having a life cycle of its own. It's born, it matures, it dies also. Born, mature and dies also. But if we are responsible for giving them chemicals, with the help of chemicals, these cells can also be sent back in their life cycle. Clear? So in 1997-96, when the first experiment of cloning was conducted at Rochester Institute, a sheep called Dolly was born by this method. And after this, what we do is we select one cell. This is somatic cell. On the other hand, we are responsible for getting from the egg provider. We are extracting nucleus. The egg cell, not nucleus, the egg. And we are removing the nucleus from it. So we are making it enucleated. So this is the egg from where the nucleus has been removed. Clear? And then what we do is transfer the nucleus of the somatic cell to the egg. Now since this is called as what? This is called as somatic cell nucleus transfer. Somatic cell nucleus transfer. So cloning is based on what? It is based on somatic cell nucleus transfer. So when it is transferred from somatic cell to nucleus, when it's transferred from the somatic cell to the egg, then the egg is just like a fertilized egg. Why? Because in somatic cell, just like a fertilized egg, 23 pairs of chromosome would be present. But in egg cell, we can see only half the number of chromosome is present. Only half the number of chromosome is present. Now this would be now inserted into put inside the womb of the surrogate mother. The baby that is born is a clone. And clone of whom? The clone of donor. But then I say that it's a clone of donor but not exactly the same. And why not exactly the same? Because mitochondria has not been transferred. Only you have transferred what? Only you have transferred the nucleus. And we can see that only nucleus has been transferred but mitochondria has not been transferred. Mitochondria also, here is the mitochondria which is belonging to the egg provider. Here is the mitochondria. Clear? Now mitochondria has the DNA of the egg provider. So it won't be exactly the same. But in one case it would be exactly the same. And what is that? When the egg provider and the donor happens to be the same person, then it would be exactly the same when the egg provider and the donor is the same person. Clear? Is the same person. Now this method is called as what? Somatic cell nucleus transfer. Somatic cell nucleus transfer. Fine. Clear? Now this is called as cloning. Now we were responsible for reverting them back in their life cycle. Why? The somatic cells. Because all the earlier experiments of cloning which we conducted by directly transferring the nucleus from the somatic cell to the egg cell, all resulted in failure. Why? Because nucleus, that is the egg, is in the initial phase of life. See, mitochondria is carrying also genetic material which is DNA. It is also the powerhouse of the cell. Powerhouse of the cell. These are two important functions of mitochondria. And out of the 20,000 genes that we have, 37 genes are determined by mitochondrial DNA. The history of mitochondria is what? That it is a bacterial cell. So it has given its self-existence and combined with the cells of the other organisms as such. Clear? Otherwise, it is a bacterial cell. Now this is called as what? This is called as somatic cell nucleus transfer, and this is how cloning is conducted. Clear? Now let's pick up the topic of what? The topic of GM crops, because genetically modified mustard is much in the news. and also genetically modified. Another crop is also in the news, which is called as BG2 RRF cotton. So let's see what is BG2 RRF cotton, which the government is considering for giving approval. So let's cover up that also briefly. Fine. Now, when we talk about genetic engineering, we say that genetic engineering is the process. Yes, धारा मस्टर्ड. धारा मस्टर्ड तो मान लो कि पूछे ना पूछे, लेकिन BG2 कॉटन भी है, थोड़ा सा जानना. फाइन. और धारा मस्टर्ड बनता है इस बार क्योंकि it has been given approval for cultivation. So maybe that they ask you question. Clear? Cold coffee, tea, coffee, drink. Okay. I'm having everything. I'm having cold also drink and having tea also. There's water also, lukewarm water also. This is also warm. This is not cold as such. Fine. I'm having everything for me. And I'll continue like this. अब देखो, genetic engineering का मतलब क्या है? Genetic engineering means that after the double helix structure of DNA was given by Watson and Crick in 1953, we can see that it became possible for the scientists to alter the genetic makeup of organism by either deleting an existing gene from it or by endowing an exotic gene into it. मान लो कि एक जीन हटा सकते हैं और दूसरे जीन को वहां पर ला सकते हैं. क्लियर? दूसरे जीन को ला सकते हैं. फाइन. Yes, that's Environment Protection Act of 1986, GEAC, that is Genetic Engineering Appraisal Committee. Now, we can see that either we can delete an existing gene or we can bring a new gene into it. Clear? Exotic gene into it. So this method of alteration of genes is called as genetic engineering. It's also called as genetic manipulation, or it is also called as recombinant gene technology. Clear? So genetic engineering को क्या कहते हैं? Genetic engineering is called as what? It's called as genetic manipulation, and is also called as recombinant gene technology. Recombinant gene technology. और जो resultant gene होगा उसको क्या कहेंगे? Thank you so much. Thank you so much. Fine. अब थोड़ा जेनेटिक इंजीनियरिंग ये GM क्रॉप्स कर लो बच्चों, और उसके बाद थोड़ा सा CRISPR-Cas9 टेक्नोलॉजी कर लो. इतना कर लो आज. Fine. Now we can see that genetic engineering is conducted by either introducing a new gene into it or by deleting an existing gene from it. Clear? So you can see the resultant gene, this would be called also as recombinant gene technology, and the resultant gene would be called as what? It is called as recombinant gene. Resultant gene क्या कहते हैं इसे? Recombinant gene. Clear? The resultant gene is referred to as the recombinant gene. Now, when we are talking about GM, genetically modified crops, GM crops, जब कहते हैं ना GM crops are those crops in which genetic engineering has been conducted. Clear? That's why we say that GM crops are those which would be essentially having rDNA present in it. rDNA means recombinant DNA present in it. That means that genetic engineering has been conducted. So it would be having recombinant DNA present in it or rDNA present in it. GM crops. Clear? And as far as GM crops are concerned, GM crops are suitable to different climatic conditions. They have rDNA present in it. And the purpose was GM crops are developed. GM crops are also called as transgenic crops. The purpose why GM crops are developed. The purpose is to, the purpose why transgenic crops are developed, the purpose is to enhance crop production The purpose is to remove diseases associated with the crop, and the purpose is also to enhance the nutrient content of crop, which is also called as biofortification. But it's not that biofortification, all fortified crops would be transgenic in nature. ये जरूरी नहीं है कि जितने भी fortified crops हैं वो transgenic ही होंगे. Clear? प्रफुल्ल महोदय का टिश्यू कल्चर. अच्छा, कुछ तो अपने से भी पढ़ोगे तुम लोग. अच्छा चलो मैं पढ़ा दूंगा. चलो देखता हूँ. क्लियर? तो now this is called as what? This is called as GM crops or transgenic crops, and we can see that it is suitable for different climatic conditions. There are many things associated with these crops, and there are notable examples of these crops. Now, it's not essential that all fortified crops would be transgenic in nature. ये कोई ज़रूरी नहीं है कि all जितने भी fortified crops हैं वो transgenic ही हों. Now, as far as GM crops are concerned, we can see the first GM crop which was cultivated was called as genetically modified tobacco. This was the first GM crop to be cultivated. Clear? Genetically modified tobacco. But the first GM food crop which was cultivated was genetically modified tomato, and this was called as what? It was called as flavor saver. Flavor saver, not VOUR. Now this is how it is written, flavor saver. There are numerous examples of GM crops in India. Clear? CRISPR-Cas9 टेक्नोलॉजी भी, गोल्डन राइस भी, सब करा दूंगा. जस्ट वेट करो कुछ थोड़ा सा. हाँ, हमारे टीचर तो अभी तक ICU में चले गए होते. ओ माय गॉड. दो बार सो के उठ भी गया. वेरी गुड. फाइन. आई लव यू ऑल. ना देखो, इसमें कई सारे एग्जाम्पल्स हैं जिसमें क्वेश्चंस पूछते हैं और उन एग्जाम्पल्स को जानना ज़रूरी है. नाउ, नॉर्मल राइस हैज़ अ डिफिशियंसी ऑफ व्हाट? इट हैज़ अ डिफिशियंसी ऑफ बीटा कैरोटीन. नॉर्मल राइस में नहीं है. नाउ व्हाट इज़ बीटा कैरोटीन? बीटा कैरोटीन व्हेन इट एंटर्स आवर बॉडी, जब ये हमारे बॉडी में एंटर करता है बीटा कैरोटीन, इट गेट्स कन्वर्टेड इनटू रेटिनॉल. और रेटिनॉल क्या है? रेटिनॉल इज़ नथिंग बट विटामिन ए. तो इसका मतलब हुआ कि नॉर्मल राइस हैज़ अ डिफिशियंसी ऑफ विटामिन ए. विटामिन ए उसमें है ही नहीं. क्लियर? सो व्हाट वी कैन सी इज दैट नॉर्मल राइस हैज़ अ डिफिशियंसी ऑफ विटामिन ए. सो the two scientists, Peter Berger and Ingo Potterkins, they were two scientists, Peter Berger and Ingo Potterkins. Now they were responsible for developing a variety of rice, and this variety they took gene from other organism, and this variety they called as what? They called it as golden rice. So this was called as what? This was called as golden rice. Clear? Referred to as golden rice. And why? Because they took what? They were responsible for taking those genes which would be helping in supplementing beta carotene. And since the color of beta carotene is golden, that's why the name golden rice. Since the color of beta carotene is golden, that's why the name is golden rice. Now, the second kind of variety is called as what? The second kind of variety is called as super rice. Super rice. So rice not only has a deficiency of beta carotene, इसमें मात्र beta carotene की ही deficiency नहीं होती है. It has a deficiency of some other things. For example, it has a deficiency of not only beta carotene but also zinc and iron. So iron and zinc are also missing. They are less in rice. And for this purpose, we can see. For this purpose, we can see that scientists of Italy developed a variety of rice, which is called as what? Which is called as super rice by genetically genetic engineering, and the iron content was enhanced four times, and zinc content was enhanced twice. So this transgenic variety of rice is called as what? It is called as super rice. Now it's not available for me and you. It's available for those areas where the children suffer from malnutrition and they consume only and only rice. For them, this is essential. Clear? For them, it is essential. You can see that in India, school children, they suffer from anemia. And that's why for them, a type of rice has been developed. And that type of rice is called as what? That type of rice is referred to as, for them, that is referred to as fortified rice. Fortified rice means there's no transgenic mechanism in it. तो उन्होंने क्या किया है कि राइस के ब्रोकन राइस को लिया है, ब्रोकन राइस को क्या किया है, इसको they have turned it into rice flour, mixed with it rice flour and mixed it with a number of nutrients and vitamin to prepare a dough, and this dough has been passed through. And this, thank you Vishnu. Now this dough has been passed through what? This has been passed through an extruder machine so that it can be cut in the shape of rice grains, और उसे dry किया है and then mixed with normal rice in the ratio of 1:100, and this is called as what? This is called as the this is referred to as what? It is fortified rice. But in fortified rice, which has been developed by IIT Kharagpur and also the DBT, no, we can see no transgenic mechanism has been used. So it's not necessary that any kind of fortification would be essentially including transgenic mechanism. But in India, there are three varieties which are much debated varieties. Now, first variety is called as what? First variety is called as BT cotton. Remember that BT cotton is also called as what? It is called as BG3 cotton. BG3 cotton. It's also called as BG3 cotton. Now, cotton suffers from what? Cotton suffers from a disease. And that's called as what? That's called as bollworm disease. Cotton is suffering from a disease which is called as bollworm disease. So, a company called, and because of which the production of cotton happens to be low. So, a company called Monsanto, and that's a US company, developed a variety of cotton which is called as what? Which is called as BT cotton. Developed a variety of cotton which is called as BT cotton. And how this cotton was developed? This cotton was developed by taking genes from soil bacterium, and this soil bacterium is called as Bacillus thuringiensis. इसे क्या कहते हैं? Bacillus thuringiensis, BT. इसीलिए इसका नाम पड़ा BT. So the genes which were taken from this bacterium included what? Cry1AC, Cry1AB, Cry9C. So all these types of genes. Now these genes, when we say Cry1AC, Cry1AB, Cry9C, all these genes are what? Now, all these genes are what? All these genes are, इस जीन को देखो, ये जीन क्या है? All these genes are called as protoxins. Protoxin का मतलब देखो, दो शब्द हैं इसमें, protein and toxin. Protoxin means protein and toxins. It means that these genes are what? These genes are essentially proteins, but they would be converting into toxic substances. Clear? So these genes are essentially proteins, but they would be converting into toxic substances when the environment is altered. और कौन से environment में जाकर ये activate होंगे? So the environment is altered. Now they would be getting converted into toxic substances. They are protein, and they are getting converted into toxic substances when the environment is altered. And we can see these proteins would be altered in what? It would be changing in alkaline environment into toxins. Alkaline environment into toxins. तो ये मान लो कि suppose the bollworm disease is caused by insect. मान लो ये insect है which is responsible for causing bollworm disease. Clear? Now the gut of the insect is alkaline in nature. गट जो है इंसेक्ट का वो अल्कलाइन है, तो जैसे ही एंटर करेगा उसके गट में, थैंक यू हिमांशु। ना अब देखो रविकांत, थैंक यू ऑल ऑफ यू। देखो जैसे ये जीन जो है क्राइजीन ये एंटर करेगा इस गट में इंसेक्ट के, व्हाट विल हैपन? इट वुड बी गेटिंग, और ये गट कैसा है? गट है अल्कलाइन। हमारे गट कैसे हैं? हमारे गट है एसिडिक। क्लियर? तो हमारे गट में तो ये कन्वर्ट नहीं होंगे टॉक्सिक सब्सटेंसेस में, लेकिन ये इंसेक्ट के गट में जाकर कन्वर्ट हो जाएंगे। क्लियर? ये इंसेक्ट के गट में जाकर कन्वर्ट हो जाएंगे। थैंक यू सो मच। अब थोड़ा सा इधर ध्यान करो। एंड इट वुड बी कन्वर्टिंग इनटू टॉक्सिन्स, और जैसे ये टॉक्सिन्स में कन्वर्ट करेंगे तो ये गट के जो वॉल है ना इसको पोरस बना देंगे, बिकॉज ऑफ विच द इंसेक्ट डाइज। द वॉल्स ऑफ द गट वुड बी मेड porous because of which insect would be dying. Clear? Because of which the insect dies. And that's why we are saying that this gene is protoxin, because it was a protein which was converted into toxic substances. Clear? That's how this works. Fine. Now this is your BT cotton. So BT cotton was given approval. It was cultivated in India on a large scale, and when it was cultivated on a large scale, there were allegations also that it reduced the fertility of soil, because when it is converted on a large scale, it is responsible for reducing the fertility of the soil. Then the thing is what? That if you are talking about BT cotton, 70% of cotton in India is nothing but BT cotton. So the allegations were wrong also, because the allegations were it caused rashes on the skin of farmers who cultivated it. It was responsible for reducing the fertility of the soil. और उसमें allegation था कि इसमें टर्मिनेटर जीन टेक्नोलॉजी भी यूज़ होता है। टर्मिनेटर जीन टेक्नोलॉजी का मतलब है कि टर्मिनेटर जीन टेक्नोलॉजी अगर किसी में यूज़ हुआ, if it is used in any suppose seed, then the seeds would be available for one generation only. It won't be available for second generation cultivation. So it would be available for one generation only. Clear? So you cannot take seeds from the first generation and cultivate it that the second generation would be present. So no, it's not there. Thank you, Dr. Prateek. Love you too. So it's not that it is possible. Fine. Now see, but these allegations were baseless, but these allegations were made. But in the year 2009, we can see we developed another variety, and this variety was also given approval by GEAC, that is Genetic Engineering Appraisal Committee. Now this is called as what? This is called as BT brinjal. Brinjal suffers from a disease, and the disease that it suffers from, yes, I'll come to Delhi before taking the premix. You are the best teacher. Thank you so much, Kavirana. Yes, so that the companies can get profit. Now just wait. Let's cover up this. Now see, BT brinjal. BT brinjal was given approval in the year 2009 by the GEAC, that is Genetic Engineering Appraisal Committee. And brinjal suffers from a disease which is called as what? It is called as fruit and shoot borer disease. Borer disease. Clear? Fruit and shoot borer disease, because of which production of brinjal happens to be less. But as far as brinjal is concerned, a company called Mexico was responsible for developing a variety of brinjal, and this variety of brinjal is called as what? It is called as BT brinjal. And the same gene from same bacterium was taken, Bacillus thuringiensis. The same gene for same bacterium was taken for cultivation, for making this transgenic variety. And what happened was that when the same gene from the same bacterium was taken, and this was approved by the GAC, but the problem was that a number of organizations were against the cultivation of BT brinjal. For example, Kheti Virasat Mission, which was headed by Dr. Umendrat. For example, Navdanya is headed by noted environmentalist Dr. Vandana Siva. For example, Gene Campaign was headed by Dr. Suman Sahai. So all these organizations were against the cultivation of BT brinjal. Please, all these organizations were against the cultivation of BT brinjal. And that's why the Mr. Jairam Ramesh, that was the then Environment Minister, was responsible for imposing a moratorium, a ban on the cultivation of BT brinjal at that time. And had it been not imposed, it would have become the first transgenic food crop to be cultivated in India. Recently, we can see Government of India was responsible for giving approval for the cultivation of GM mustard. Clear? GM mustard. Clear? Now we can see GM mustard, and this has been given approval. Now if we talk about mustard cultivation, we can see mustard suffers from some problem. So mustard cultivation, if suppose we are talking about mustard cultivation, first problem in mustard cultivation is that mustard is a self-pollinating crop, and that's why the hybrid varieties of mustard are not possible. That's a self-pollinating crop. Hybrid varieties of mustard are not possible. Second problem is what? That when you cultivate mustard, in between the mustard, weeds developed. Now, if you are responsible for using a herbicide, and the herbicide commonly used in India is called as glufosinate, which is developed by Bayer. That's the herbicide commonly used in India. So if we use glufosinate for this purpose, what will happen due to glufosinate? When it is sprayed on the mustard field, not only the weeds get damaged, but also the mustard gets damaged. So what we have to do is, these are two problems in mustard cultivation. So we have to deploy manual laborers for this purpose. And if you have deployed manual laborers for this purpose, then what will happen is that there would be a problem. And the problem is, it is a costly affair. Yes, our native vegetables are more delicious. Are those were against? Yes, those organizations were against the cultivation of BT brinjal. Now, that is the problem with mustard, that mustard cultivation becomes costly affair because of manual laborers being deployed for this purpose. Clear? Manual laborers being deployed for this purpose. So a group of scientists of Delhi University, and this group of scientists of Delhi University was headed by Dr. Deepak Pentel. Dr. Deepak Pentel. They developed a variety of mustard which is called as what? Which is called as GM mustard. It is also called DMH11. That is Dhara Mustard Hybrid 11. So that's called as Dhara Mustard Hybrid 11, or which is called as DMH11. And for this purpose, now this is what? This is a HT variety. HT variety means herbicide tolerant variety. And herbicide tolerant variety means that yes, it is approved by GAC, clear, but GAC has ordered the cultivation of this mustard, that it can be cultivated. Now, Dhara mustard, so this is a HT variety, that is herbicide tolerant variety of mustard. It means that if you spray herbicide, the weeds would be getting damaged but not the mustard. But for developing this variety, what we have done, we have taken two variety of mustard. Now, one variety of mustard is called as what? One variety of mustard is an Indian variety, and that variety is Varuna. So that's an Indian variety. Clear? And the second variety of mustard that we have taken is called as what? It's called as Hera. And this is what? This is an East European variety. East European variety of mustard. Clear? And then from a bacterium which is called as Streptomyces. Streptomyces, we have taken a gene called Barstar. First is Barnase. Now we have introduced this gene Barnase into Varuna to make it what? To make it male sterilized. Male sterilized means the male part of the flower of Varuna won't be functioning. The female part would be only active. And then from this gene, again, a gene has been taken which is called as Barstar. And this has been used in Hera. And the purpose is to make it female sterilized. So means that male part would be only functioning, female part won't be functioning. Clear? And then some additional genes have been introduced in Varuna. Additional genes. And these additional genes have been introduced in Varuna to make it HT in nature. That is herbicide tolerant in nature. And then we can see that cross pollination has been conducted between the two, because mustard was a self-pollinating crop. So cross pollination has been conducted between the two. Cross pollination has been conducted between the two, and the resultant variety is called as what? It is called as DMH11. Dhara mustard hybrid 11, which is a HT variety. HT variety means herbicide tolerant variety. Clear? And if you cultivate this mustard and then on the field you are responsible for clear? Thank you so much. SK, thank you so much. Fine. So let's complete this. Now this is called as DMH11 and this is HT variety. Now if you cultivate this variety on the field, and if this variety is cultivated on the field and then glufosinate is sprayed, so the weeds would be getting damaged but not the mustard as such. So the weeds are going to get damaged, but it's not the mustard which would be getting damaged. So this is called as GM mustard. Is that clear? Fine. GM mustard. Yes, Shivam Saho, how you continue energy? My energy is you all people. Fine. Let's deal with another thing, and maybe that comes in examination also. And that is called as the GAAC is also considering giving approval to another type of cotton. And this cotton is now presently owned by Bayer. It was initially made by Monsanto, but it has been purchased by Bayer. And this is also HT variety, and the HT variety of cotton is called as what? It is called as BG2RRF cotton. So let's see what is BG2RRF cotton. This is one thing, but another variety of cotton is called as what? BG2RRF. देखो जब कहते हैं ना BG3, BG3 is nothing but BT cotton. But BG to RRF cotton means Bolgat 2. And RRF means round, ready, flex. Round, ready, flex, RR2. Now this cotton is today owned by Bayer. किसका है ये? Bayer. And this Bayer has purchased it from where? Purchased is from Monsanto. And this is what? This is HT variety of cotton. So we have HT variety of mustard, herbicide tolerant variety of mustard. Now we have herbicide tolerant variety of cotton. So the GAC is considering giving approval to this variety of cotton. And as far as this variety of cotton is concerned, it consists of a gene. Now the gene are Cry1AC, 1AC, Cry2AB, 2AB. Now all these genes have been taken from where? It has been taken from soil bacterium. But in this type of cotton, that is BG2 RRF, another gene also has been taken, and that is called as CB4 EPS PS. So that is taken from an agrobacterium. So remember the genes, agrobacterium. Now these are some of the varieties that you have to know. BT cotton, then BT brinjal, then GM mustard, and also BG2 cotton. Clear? And for cultivating BG2 cotton, we are using another herbicide of, we would be using another herbicide of Bayer, and this is called as glyphosate. Glyphosate was earlier associated with farmers' suicide in India. Why? Because it was not allowed for the farmers to be used. In some, only in some of the cultivation it was allowed. Otherwise, it was not allowed to be used by the farmers, glyphosate. Clear? But when farmers used it, they sprinkled it, they inhaled it also. And this was responsible for death among the farmers because farmers suffered from cancer. Clear? Now, we'll discuss another thing in biotechnology, and that's also a question which is asked, and that is called as what? It is called as CRISPR-Cas9 technology. Clear? So there's a term which is called as gene therapy, and there's a term which is called as CRISPR-Cas9 technology. Cas9 technology. So let's see what is CRISPR-Cas9 technology and what is glyphosate. Glyphosate is what? Agrobacterium is, that is the bacterium which is derived from the agricultural field. What is glyphosate? Glyphosate is nothing but a type of herbicide which is used by Bayer, developed by Bayer. It was earlier with Monsanto, it has been purchased by Bayer. Pesticide नहीं है, ये herbicide है. You have covered pesticide ban and illegal trade. No, I have covered. Sir, have you covered pesticides ban and illegal? No, I haven't covered that. I have covered some of the important things only because I cannot cover all those things. But now let's see what is called as gene therapy and also what is called as CRISPR-Cas9 technology. Now, when we talk about gene therapy, now this gene therapy was developed by W. French Ederson. W. French Ederson. And as far as gene therapy is concerned, the purpose of gene therapy is to remove inherited disorder in a person. Clear? The purpose, thank you, Samksha. Yes, gene editing tool I'll discuss just now. Now, the purpose of gene therapy is what? The purpose of gene therapy is to develop. The purpose of gene therapy is to remove inherited disorder in a person. It was developed by W. French Ederson. And how it is removed? First of all, what happens is that Yes, Saurav Verma, is this class is literally going on at 1:46 a.m. at night, definitely. सो के उठ गए सौरभ वर्मा क्या, कि सोए नहीं? Good to see you. Good to see you, Samiksha. Fine. And hats off to you also. You also. Because, now see, W. French Ederson. It was developed by W. French Ederson, and the purpose is what? The purpose is to remove inherited disorder in a person. मान लो अगर किसी में inherited disorder है, तो उस inherited disorder को हटाने के लिए क्या करते हैं? जीन थेरेपी का इस्तेमाल करेंगे. Clear? Now, how that is done? First of all, what we do is that we are responsible for identifying the faulty gene. Faulty gene को identify कर लेते हैं. Clear? Faulty gene would be identified. And then, if there is a genetic disorder, there would be a faulty gene responsible for it. Then we can see that normal version of this gene is developed in the lab. So, we can see normal version of this gene is developed in the lab. Clear? Normal version is developed in the lab. And faulty gene is identified. Normal version is developed in the lab. But normal version is developed by cloning. ठीक है, मान लो अब नॉर्मल वर्जन डेवलप आपने कर ही लिया जीन का. नाउ व्हाट यू डू इज दैट यू आर टेकिंग होस्ट सेल्स फ्रॉम द बॉडी ऑफ द पेशेंट. होस्ट सेल्स लेंगे किसके बॉडी से? पेशेंट के बॉडी से. क्लियर? होस्ट सेल्स वुड बी ओके. गेट वेल सून सौरभ. Host cells would be selected from the body of the patient. नींद आने नहीं लगी अभी, रुक जाओ कुछ, रुक जाओ. हाँ सौरभ, ये 13 आवर्स में कंटिन्यूस पढ़ा रहा हूँ बेटे. मैंने प्रॉमिस करके आया वाइफ को कि आज सारे रिकॉर्ड तोड़ कर आऊँगा घर. फाइन. नाउ होस्ट सेल्स आर सिलेक्टेड फ्रॉम द बॉडी ऑफ द पेशेंट, एंड देन इन दीज सेल्स द नॉर्मल वर्जन जीन्स आर ट्रांसफर्ड. मान लो इसमें फिर नॉर्मल वर्जन जीन्स जानते हैं. तो जैसे ही होस्ट सेल्स में आपने नॉर्मल वर्जन जीन्स को डाला, नाउ दीज आर कॉल्ड एज व्हाट? अब इसे कहेंगे हम लोग डॉक्टर्ड सेल्स. डॉक्टर्ड सेल्स. और इन डॉक्टर सेल्स को यू वुड बी रिटर्निंग बैक टू द बॉडी ऑफ द पेशेंट थ्रू इंजेक्शन. So when you are returning back this to the body of the patient through injection, the disorder is gradually removed. The disorder is gradually removed. Fine. The disorder is gradually removed. Clear? लेकिन देखो प्रॉब्लम इसमें क्या है कि डिसऑर्डर तो ग्रेजुअली रिमूव होता है. 2 सेल्स हैं आपके बॉडी में ब्रॉडली. एक तो आपके रिप्रोडक्टिव सेल्स हैं और दूसरा क्या सेल्स है? दूसरा सेल्स जो है आपके बॉडी में वो कहा जाता है सोमेटिक सेल्स. तो रिप्रोडक्टिव सेल्स के बाद जितने भी सेल्स आपके बॉडी में हैं सारे सोमेटिक सेल्स ही हैं. क्लियर? सो वी कैन सी. Thank you a lot. Thank you a lot. I love this. यार क्या लाजवाब पढ़ाते हैं. 20 मिनट की क्लास की है, सर का फैन हो गया. आपने सिर्फ 20 मिनट की क्लास की है. अच्छा, अब इसे पढ़ के ही जाओ. कुछ देर और मैं पढ़ाऊंगा और इसको इसे पढ़ के ही जाओ. क्लियर? फाइन. फिर Now see, you cannot replace all the cells by gene therapy. देखो इसमें क्या है ना कि reproductive cells जो हैं आपके, इसको छोड़कर जितने भी cells हैं आपकी body में, they are called as somatic cells. So what happens is that when gene therapy is conducted, gene therapy would be conducted for only somatic cells. हो सकता है कि आपके reproductive cells से फिर वो disorder चला जाए next generation में. again the disorder would be returning back in the next generation. Why? Because gene therapy is conducted for somatic cell only. It's never conducted for reproductive cells. Clear? So it would be carried by the reproductive cells to the subsequent generation, and again gene therapy would be conducted to resurrect that. फिर से उसको जीन थेरेपी कंडक्ट करना पड़ेगा ताकि हम लोग रीसरेक्ट हो जाएं. क्लियर? नाउ देखो, अब this is the case that this is gene therapy. Now the problem with gene therapy was what? That it can be used for only somatic cells, but it cannot be used for reproductive cells. अब इसे हटा दो दिमाग से, नॉट दिमाग से मत हटाओ, यहाँ से हटा दो, बोर्ड से. Now CRISPR-Cas9 टेक्नोलॉजी के साथ क्या है? CRISPR-Cas9 technology is a technology in which we would be responsible for gene editing. We would be responsible for gene editing. And where? In the reproductive cells. Reproductive cells में हम genes editing कर सकते हैं. Somatic cells में तो कर ही सकते हैं. Embryos में कर सकते हैं gene editing. मतलब जेनेटिक इंजीनियरिंग आप एम्ब्रियोज में भी कर सकते हैं और जेनेटिक इंजीनियरिंग आप रिप्रोडक्टिव सेल्स में कर सकते हैं. तो इसे क्या कहते हैं? इसे हम लोग कहते हैं क्या? CRISPR-Cas9 टेक्नोलॉजी. नाउ CRISPR-Cas9 टेक्नोलॉजी स्टैंड्स फॉर व्हाट? देखो, CRISPR का मतलब है क्लस्टर्ड रेगुलरली इंटरस्पेस्ड palindromic repeats. That is clustered, regularly interspaced palindromic repeats. तो इसमें दो टूल हैं, and the tool is what? You cannot replace all somatic cells inside the body. Now this is called as what? It's called as clustered, regularly interspaced palindromic repeats. Now CRISPR-Cas9 technology has two tools. दो tools है इसमें. There are two tools present in CRISPR-Cas9 technology. Now the first tool which is present in CRISPR-Cas9 technology is what? It is a sequence of RNA. Clear? Now sequence of RNA, मान लो ये sequence है RNA का. Now this sequence of RNA goes and binds with the sequence of DNA which needs to be removed. So there are two tools present in CRISPR-Cas9 technology. One is sequence of RNA which goes and binds with the sequence of DNA which needs to be removed. So that's one thing. And Cas9 is what? Cas9 is acting as a molecular scissor. Cas9 क्या करता है? इसे काटता है. तो पहले ये आरएनए का सीक्वेंस जाकर डीएनए के सीक्वेंस से बाइंड करेगा, जुड़ जाएगा, क्योंकि रिप्रोडक्टिव सेल्स में डीएनए को आप आसानी से हटा नहीं सकते. सो दिस वुड बी गोइंग एंड बाइंडिंग विद द सीक्वेंस ऑफ डीएनए व्हिच नीड्स टू बी रिमूव्ड, एंड देन Cas9 इज अ टाइप ऑफ एंजाइम व्हिच वुड बी स्ट्रिपिंग ऑफ, जो उसे जाकर काट देगा. क्लियर? सो Cas9 इज एक्टिंग एज व्हाट? मॉलिक्यूलर सीजर्स. ये पीटी में आपको पूछा है. Clear? Cas9 is acting as what? Molecular scissor, and the reproductive, and we can see gene editing can be conducted. Clear? So Cas9 is acting as a molecular scissor. Clear? So we can see that this is called as CRISPR-Cas9 technology. ठीक है, अच्छी बात. Now CRISPR-Cas9 टेक्नोलॉजी से आप कर क्या सकते हो? दिस टेक्नोलॉजी वॉज डेवलप्ड बाय, अब ये याद रखना है कि एक सीक्वेंस है, दिस इज सीक्वेंस ऑफ आरएनए, और दूसरा क्या है? Cas9 इज अ टाइप ऑफ एंजाइम विच एक्ट्स एज अ मॉलिक्यूलर सीजर. बट CRISPR-Cas9 टेक्नोलॉजी वॉज डेवलप्ड बाय whom? CRISPR-Cas9 टेक्नोलॉजी वॉज डेवलप्ड बाय डॉडना एंड कार्पेंटियर. दो साइंटिस्ट थे, इन्होंने इसको डिस्कवर किया था, डॉडना एंड कार्पेंटियर. And this was developed by Doudna and Carpentier, and they were conferred the Nobel Prize in the year 2020 for this technology. Because CRISPR-Cas9 technology has wide application in the field of agriculture. एग्रीकल्चर के फील्ड में अगर देखो तो इसका वाइड एप्लीकेशन है इन द फील्ड ऑफ एग्रीकल्चर. क्लियर? एग्रीकल्चर के फील्ड में दिस इज फॉर द पर्पस ऑफ व्हाट? दिस इज फॉर द पर्पस ऑफ इनहेंसिंग क्रॉप प्रोडक्शन. स्पेसिफिक एग्जाम्पल जीन थेरेपी. देखो कोई भी इनहेरिटेड डिसऑर्डर है, अगर उसको रिमूव करते हैं तो जीन थेरेपी इसे करते हैं जनरली. अब आप लोग ये ना बोले कि आपको इतनी मेहनत करने की बाद भी सब याद कैसे है, मैं सब भूल जाता. सर यू गिव स्पेसिफिक एग्जांपल्स ऑफ जीन थेरेपी. देयर इज नो स्पेसिफिक एग्जांपल एज सच, बट देखो CRISPR-Cas9 टेक्नोलॉजी मैं रिपीट कर देता हूँ. हाँ, एथिकल इश्यूज में भी उसके बारे में बता सकता हूँ. देखो CRISPR-Cas9 टेक्नोलॉजी में क्या है ना, एक बार मैं रिपीट करता हूँ. दो टूल्स हैं. एक टूल्स क्या है कि मान लो ये सेगमेंट है डीएनए का जिसे आपको हटाना है, तो देयर आर टू टूल्स. एक है सेगमेंट ऑफ आरएनए. नाउ दिस सेगमेंट ऑफ आरएनए व्हिच यू आर हैविंग वुड बी बाइंडिंग विद द सेगमेंट ऑफ डीएनए व्हिच नीड्स टू बी रिमूव्ड. क्लियर? यस. और ऐसे ही आप इसे काट नहीं सकते बिकॉज दिस इज रिप्रोडक्टिव सेल. क्लियर? You cannot cut off. So this is a segment of DNA which goes and binds with the segment of RNA, DNA which needs to be removed. And for removing this, you are using Cas9 because Cas9 would be acting as molecular scissors. तो एंजाइम जो Cas9 है वो क्या करता है, मॉलिक्यूलर सीजर्स के रूप में काम करता है और उसे काट देगा. और यहाँ पर आप नई जीन को इंट्रोड्यूस कर सकते हैं या डिलीटिंग एग्जिस्टिंग जीन को डिलीट कर सकते हैं, न्यू जीन को इंट्रोड्यूस कर सकते हैं. तो ये है CRISPR-Cas9 टेक्नोलॉजी. क्लियर? अच्छी बात है. लेकिन देखो when you are talking about CRISPR-Cas9 technology, I have said that Dodna, Carpentier developed it. They were conferred the Nobel Prize also because it has vast application in the field of agriculture because it was responsible for biofortification, removing diseases of plants, enhancing crop production. ये सारे थे उसके benefits. But CRISPR-Cas9 technology has been also criticized. क्योंकि इससे क्या करते हैं, ना केवल आप रिप्रोडक्टिव सेल, फर्स्ट ऑफ ऑल द प्रॉब्लम इज व्हाट दैट इफ you are responsible for thank you so much, if you are responsible for any alteration in reproductive cell, this alteration in reproductive cell can go on for subsequent generation. अगर मान लो reproductive cells में आप किसी तरह के alteration करते हो तो ये alteration तो subsequent generation तक पहुंच जाएगा. Clear? subsequent generation तक पहुंच जाएगा and you never know the repercussions. The second thing is what? That if you have suppose conducted a genetic alteration in the embryos and you are deliberately responsible for conducting genetic alteration in the embryos, you never know that when the embryos would be growing into a developed adult, he or she may not like the genetic alteration which you were responsible for. ये भी तो हो सकता है. Clear? That may not be in the fashion also. पेरेंट्स हैं आप, आपने CRISPR-Cas9 टेक्नोलॉजी की मदद से क्या किया कि अपने बेबी को डिजाइन कर दिया. बिकॉज इन दिस कंटेक्स्ट द कॉन्सेप्ट ऑफ डिजाइनर बेबी इज ऑल्सो गिवन. डिजाइनर बेबी का मतलब है कि द पेरेंट्स दे विश देयर बेबीज टू बी डिजाइन इन सच ए मैनर कि ऐसे ऐसे फीचर्स उसमें होने चाहिए. सो CRISPR-Cas9 टेक्नोलॉजी इज यूज्ड फॉर डिजाइनर बेबी ऑल्सो. बट हाउ इट इज यूज्ड फॉर डिजाइनर बेबी, देखो कैसे यूज होता है डिजाइनर बेबी में, ये भी जान लो. First, when we are talking about designer baby, we can see that the mother is given a hormonal injection. मदर को क्या करते हैं, हार्मोनल इंजेक्शन देते हैं ताकि एक से ज़्यादा ओवम वो रिलीज करे एक समय में. मान लो 4 ओवम रिलीज किया उन्होंने, मदर ने, एक समय में. क्लियर? और ये चारों का इन विट्रो फर्टिलाइजेशन करते हैं, या तो फादर के स्पर्म से फर्टिलाइजेशन करेंगे या सिलेक्टेड डोनर्स के. तो चलो ये IVF किया लैब में, इन विट्रो फर्टिलाइजेशन, लैब में फर्टिलाइजेशन हुआ. और इसमें क्या है कि 4 एम्ब्रियोज आपने बनाए. यू हैव क्रिएटेड 4 zygotes बाय फर्टिलाइजिंग. Now we scan these zygotes. अब इसको स्कैन करते हैं कि जो कैरेक्टरिस्टिक मदर या फादर में उन्हें विश किया है, they are yes blue eyes, red or green. Now जो कैरेक्टरिस्टिक ये मदर एंड फादर ने विश किया है वो कैरेक्टरिस्टिक इसमें है कि नहीं, ये देखेंगे. क्लियर? Those characteristics are present in this embryo or not, zygote or not. And मान लो अगर एक में है तो this would be selected and the remaining would be discarded. Now here CRISPR-Cas9 technology would be used to enhance the characteristic so that it would be meeting the requirement of parents. Clear? To enhance the characteristic so that it would be meeting the requirement of parents. And we can see that after CRISPR-Cas9 technology is done, then this embryo would be planted in the womb of surrogate mother. The baby which is born is nothing but designer baby. देखो ये baby है. This is designer baby. बच्चे हैं ना फेंकते हैं हाथ पैर अपना. So this is called as designer baby and this is how designer baby is born. So with the help of CRISPR-Cas9 technology, the first designer baby was born in Los Angeles in the year 2012. This was how designer baby was born. But as far as designer baby is concerned, देखो इसमें एक पॉजिटिव पर्सपेक्टिव भी है, एक नेगेटिव पर्सपेक्टिव भी है. पॉजिटिव पर्सपेक्टिव क्या है? कॉन्स्टेंटली द एम्ब्रियो इज बीइंग स्कैन्ड. If suppose looks like an ant, yes. No, if the embryo is scanned, if any kind of genetic disorder is present, yes, cute baby. If any kind of genetic disorder is present, it can be known to us. But the negative part of it is what? That a number of embryos have been discarded also. What about them? They are life forms, so should not have been discarded. Clear? Should not have been discarded. So that's called as designer baby. Also we can see that there was a Chinese scientist, his name was Jian. So Jian was responsible for what? Jian was responsible for developing with the help of CRISPR-Cas9 technology, developed twins which shows innate resistance to, which shows inbuilt resistance to. What? Inbuilt resistance to, which is showing innate or inbuilt resistance to HIV. But how? But how they were responsible for developing this baby which shows innate resistance to HIV? Let's discuss that also. Now, we can see that Jian was a Chinese scientist and he was responsible for developing two twins. Name याद रखना जरूरी नहीं है, but these two twins, they had innate resistance to, that is inbuilt resistance to, inbuilt resistance to HIV. How? Now you can see that how. Now there is a, in the genetic makeup of humans, there is a gene which is called as what? This is called as CCR5. CCR5. Now CCR5 is responsible for codifying for a protein, which allows the invasion of HIV. Just remember one thing, that the genetic information, मैं अभी एक बस थोड़ा सा और पढ़ाऊंगा बच्चों, उसके बाद then we'll have not a set of twins. No, twins means two only. Now, Jian developed twins who were having innate resistance to HIV. Now, what happens? Understand this, that the information that we have in our genetic makeup, this is the information, suppose, whatever information we have in the genetic makeup. Now, it gets converted into action. It gets converted into action with the help of what? With the help of proteins. Clear? So if we are saying that 20,000 genes are present in humans, 20,000 proteins are also present, which converts the information into action. For example, in UPSC they have asked you a type of gene which is called as ACTN3. ये पूछा है आपको. Now ACTN3 gene तो present है किसमें? It is present in, definitely in humans. Clear? It is present in human, ACTN3 gene. And ACTN3 gene is present not in all humans. I do not have this gene because it is present in fast athletes. किसमें है? Fast athletes में. Theory क्या है इसका? This gene is present, ACTN3 gene, and the information is contained in this gene. But there are two types of muscles inside your body. अच्छा रुक जाओ, मैं बता रहा हूँ CCR5. Now there are two types of muscles inside our body. There one type of muscle is called as what? One type of muscle is called as fast twitch muscle, and second type of muscle is called as slow twitch muscle. मान लो कि अगर कोई काम आपको करना है जिसमें सडन आउटबर्स्ट की जरूरत हो, सडन आउटबर्स्ट की जरूरत हो, तो इन दैट केस फास्ट ट्विच मसल्स would be contracting. मान लो तो तेज दौड़ना है, तो contraction of fast twitch muscle is essential. So you need a sudden outburst, and contraction of fast twitch muscle is essential. Clear? Contraction of fast twitch muscle is essential so that it gives you a sudden outburst to run fast. And if suppose a person has to perform any work of greater endurance, then contraction of slow twitch muscle is essential. Clear? Contraction of slow twitch muscle is essential. Fast twitch muscle and slow twitch muscle. Clear? And if we are having ACTN3 gene, this gene has the information, but this information is getting translated into action with the help of a protein called actinin A. And the action which is taking place is contraction of fast twitch muscle so that a person can run fast. So the information is contained in the gene, it is getting translated into action with the help of a protein, and this protein is called as actinin A. This protein is called as actinin A. So what we can see that Jayant identified a gene which is called as CCR5. Now this codifies for a protein which allows the invasion of HIV in our body. So he used what? He used CRISPR-Cas9 टेक्नोलॉजी. देखो ये CCR5 जो है जीन हमारे जेनेटिक मेकअप में है, और ये जीन कॉडिफाई करता है एक प्रोटीन के लिए जिसके कारण HIV हमारे बॉडी में एंटर कर सकता है. So what he did was he used CRISPR-Cas9 टेक्नोलॉजी. अब Jayant ने क्या किया? CRISPR-Cas9 टेक्नोलॉजी यूज किया, and he brought a mutation in this gene. And this mutation is called as what? This mutation is called as CCR5 delta 32. This mutation is called as what? It is called as CCR5 delta 32. And because of this, we can see that the babies would be having innate resistance to, the twins would be having inbuilt resistance to HIV. HIV उन्हें होगा ही नहीं. But then this experiment of Jayant has been criticized, because you have found the remedy before the disease has taken place. Clear? You have found the remedies before the disease have taken place. And for this purpose, we can see that CRISPR-Cas9 technology was used by, and the scientists all over the world criticized also this accomplishment, saying that this would be raising the vulnerability of the child for other diseases. So this is called as what? This is referred to as CRISPR-Cas9 technology. Clear? CRISPR-Cas9 technology. Now, a brief information about the stem cells. Stem cells are those cells which are present in the youthful stage. Youthful stage means they do not undergo cell division of their own. So they are present in the youthful stage. And these cells are also called as what? These cells are also called as master cells. Clear? यस, मैं सिर्फ बेसिक ही बोलूंगा, उतने समय भी नहीं है बच्चों, अब थोड़ा इसे एंड भी किया जाए कि दीज सेल्स आर ऑल्सो कॉल्ड एज व्हाट, दीज सेल्स आर ऑल्सो कॉल्ड एज मास्टर सेल्स। इसे क्या करते हैं, मास्टर सेल भी कहते हैं। सो दैट्स कॉल्ड एज व्हाट, master cell. And as far as these cells are concerned, stem cells are concerned, they have a characteristic which is called as pluripotency. Pluripotency का मतलब है कि these cells can convert into other type of cells. Pluri मतलब many, potency मतलब the ability to transform. So these cells have the ability to convert into other type of cells also. So that's called as pluripotency. Clear? So stem cells have the characteristic which is called as pluripotency. They have the ability to transform. So this can be used for what? This can be used for making of organs. This can be used for what? Making of tissues. Making of tissues. And these cells, since they are used for making of organs, making of tissues, removing diseases also. Removing diseases also. So they are called as what? They are also called as master cells. They are also called as master cells. Clear? Stem cells are called as master cells. And we can see that clear, these cells are also referred to as master cells.